Today we're going to be looking at sex-linked patterns of inheritance. Now this does tend to be a more complicated pattern of inheritance. Up until now we've been looking at those genes that are considered autosomal. Remember that term autosomal is referring to those autosomes, those homologous pairs of chromosomes with genes for most of the traits that we're interested in. So today we're going to focus in on just those sex chromosomes. And for humans we have a pair. That pair of sex chromosomes has some of the genes that determine sex characteristics, but then we are also going to have other genes on those sex chromosomes that can determine other traits. So for example, from the lecture slides and the textbook chapter, we've got eye color in fruit flies, and that's due to a gene on the X chromosome. And so we know that on the sex chromosomes, we can have genes that can lead to other traits or other things. It's not just about the sex characteristics. For the most part, if an individual has two X chromosomes, then most likely they're female. If an individual has an X and a Y, most likely they are male. So once we have started looking at a trait that we think is sex-linked, remember what we have to do is we have to use a Punnett square. And when using a Punnett square, we're going to follow those four main steps. We're first going to figure out our parental genotypes. We're then going to model meiosis, model fertilization, and then interpret the data. It's in interpreting the data where things get a little bit more complicated with our sex-linked patterns of inheritance. So I'd like to work through an example of a sex-linked pattern of inheritance. Remember, the letter B stands for our parental generation. So if I have a female, the first thing that I need to do is I need to communicate that this gene is on a sex chromosome. So I'm going to have my two X chromosomes. My female has a heterozygous genotype. Remember, when I'm writing that genotype, after I communicate that I've got the sex chromosomes, I put a letter assigned to that sex chromosome. In this case, it's a capital letter communicating that this X chromosome has a dominant gene. On this X chromosome, I have a lowercase letter. This is communicating that this X chromosome has that recessive gene. This female is going to mate with a male. The male's sex chromosomes are X and Y, and this male happens to be hemizygous dominant. We have to use that term hemizygous because the sex chromosomes for the male are not the same. The Y chromosome is a little bit smaller, and even though it doesn't have, even though we're not focusing on the Y chromosome, it does have interesting genes, but it does not have the gene indicated by the letter A. It quite literally does not exist on the Y chromosome, so I don't put any letter on that Y chromosome. Our male has a hemizygous dominant genotype, and he is going to mate with a female who is a heterozygous genotype. So I know my parent genotypes. The next step is to model meiosis. And remember, when we're modeling meiosis, our gametes go on the top and the side. It doesn't matter if we put our females' gametes on the top or on the side, we're still going to end up with the same data in the center of the square, the same data for our F1 diploid zygote, otherwise known as the kid. So I'm going to model the separation of this homologous pair of chromosomes for my female. 50% of her gametes get the X chromosome with the dominant gene, and 50% of her gametes get the X chromosome with the recessive gene. I now need to model my male's sex chromosomes going through meiosis. 50% of my male's gametes get the X chromosome with that dominant gene, and 50% of my male's gametes will get the Y chromosome. Next step is I model fertilization, the bringing together of the pairs of chromosomes. Remember that when we are making this square, we are modeling the possibilities for just one diploid zygote from these two individuals coming together. So there, I've modeled fertilization. The next step is I need to interpret my data. Now here's where things get a little different. When I'm interpreting my data, I first have to take into account the possibility of my diploid zygote being male or female. There's always a 50% chance that my diploid zygote will be female and inherit two X chromosomes, a 50% chance that my diploid zygote would be male and inherit the X and the Y chromosome. So I need to separate my perspectives. I'm going to start with the possibility that my F1 generation, my diploid zygote, is female. I'm going to look for the genotypes that are possible, figure out the probability. And then, based off of those genotypes, the phenotypes that are possible and the probability. So looking only at the top half of my Punnett square, looking at the genotypes that are possible if my diploid zygote were female. If my diploid zygote were female, what I see is that one possible genotype is homozygous dominant, the other possible genotype is heterozygous. The possibility that my female diploid zygote has a homozygous dominant genotype is 50%. The probability that my female diploid zygote will inherit a heterozygous genotype is 50%. Now both of these genotypes will show me the dominant phenotype. So the probability that my female diploid zygote will show me the dominant phenotype is 100%. Now I have to look at the other possibility. What if my diploid zygote were male? If my diploid zygote were male, what are the possible genotypes? Based on those genotypes, what are the possible phenotypes? And then going to have to squeeze it in right next to the end, the probability of seeing those different phenotypes. So looking only at the bottom part of my inner square, I see that one possible genotype for my male diploid zygote is hemizygous dominant. Probability my male diploid zygote will receive a hemizygous dominant genotype is 50%. The other possible genotype is hemizygous recessive. And the probability my male diploid zygote will receive that hemizygous recessive genotype is 50%. Since both genotypes are possible for my male diploid zygote, I know I have the possibility of seeing both phenotypes. The probability I will see the dominant phenotype is 50%, and the probability I will see that recessive phenotype is 50%. So, what will be interesting in my F1 generation is that any time I see that recessive phenotype, I automatically know that that individual is male, because in my F1 generation, only the males will show me that recessive phenotype. And I know that that is a 50% probability that my males will show me that recessive phenotype. Hopefully seeing the data worked out in this very predictable way, hopefully this helps you to understand why when we're doing sex-linked patterns of inheritance, at least going from the parental generation to the F1 generation, hopefully this helps you understand why we will not have or see a 25% probability or a 75% probability. It's because we first have to take into account the possibility of our diploid zygote being female or male, and then that automatically means we will have a 0, 50, or 100% probability in our genotypes and phenotype data. As always, if you have any questions, please email me